Hello everyone. Now we are going to discuss about culture methods. Microbial culture is a process of growing and maintaining microorganisms under controlled laboratory conditions. Why it is necessary means to study about microbes, their characteristics and their interaction with other microorganisms. Here we can list out some of the purpose of doing culture. To isolate bacteria in pure culture, then to demonstrate their properties, to obtain a bulk amount of organism for preparation of antigen and other biochemical analysis, to determine sensitivity of antibiotics, to estimate viable counts and to maintain stock culture for long-term use. The most common microbial culture techniques used are streak culture, lawn culture, stroke culture, stab culture, pore plate culture, and liquid culture. Streak culture. It is the most widely used method to isolate bacteria. One loop full of culture is made as a primary inoculum and it is then distributed thinly over the plate by streaking it with the loop in a series of parallel lines in different segments of plates. Loop claimed and cooled between the different sets of streaks. On incubation, growth may be confluent at the site of original inoculation but becomes progressively thinner and well separated colonies are obtained over the final series of streaks. Lawn culture, this method is otherwise called as carpet culture. This method mainly provides uniform growth of bacterium and it is useful for bacteriophage typing and antibiotic sensitivity testing. Also used in preparation of bacterial antigen and vaccines. Mainly, it is prepared by flooding the surface of the plate with a liquid culture or suspension of the bacterium, pipetting of the excess inoculum and incubating the plate. Alternatively, the surface of the plate may be inoculated by applying the swab soaked in a bacterial culture or suspension. In this method, cotton swab is dipped in culture near flame, then the swab is spread with culture is swabbed on the plate. Here we can see the antibiotic sensitivity plate. This lawn culture is mainly used in antibiotic sensitivity assay. Next culture method is stroke culture. Usually stroke culture is made in tubes containing agar gland. This method is mainly employed for agglutination kind of diagnostic test. Next culture method is pore plate method. This method is mainly used for quantifying microorganisms. Here the sample is mixed with agar and poured into a plate. After incubation we can observe the colonies throughout the agar, not just the surface. This image shows us the incubated plates of serial dilution. Here we can observe the colony number decreases with serial dilution concentrations. This is colony counter. With the help of this instrument, we can easily count the colonies which is present in the incubated plates. Next kind of culture method is stab culture. It is prepared. It is mainly prepared by puncturing with a long straight charged wire in a suitable medium, such as nutrient gelatin or glucose agar. Medium is allowed to set with a tube in the upright position, providing a flat surface at the top of the medium. Employed mainly for demonstration of gelatin liquefaction and oxygen requirement of the bacterium under study. Also used in the maintenance of stock culture. Another culture method is liquid culture. In liquid culture, microbes were grown in nutrient broth. Then the tubes were allowed for a larger population and easy harvesting, which were mainly used for DNA isolation, safe flask, or in bioreactors for product yield. Thank you.